So today we'll be discussing more details about synapses and neurotransmitters, which is how neurons talk to each other, different kinds of ways they use for communication of different kinds. And this is basically how the brain organizes information, which is ultimately everything, because that's how we move, how we speak, how we think, how we feel, and well, everything that we do. All right, let's get to it. And of course, feel free to interrupt me at any moment to ask any questions or anything. And yeah, we'll just go through this. Right. So first, our today's topic is going to be, by the way, do you see the screen?
Yeah.
Okay. So today's topic is going to be signal transmission and synaptic logic. And by signal transmission, we just mean that neurons have to send each other signals, which just says, for example, hey, something is touching my skin, or hey, I'm feeling pain, or I'm feeling sad, or I want to say this, or I want to move to that room. And synaptic logic is just how all of this is handled. So let's do it. First of all, let's start by remembering what a neuron looks like. So neurons have a cell body, which is just this large spherical shape in the middle, which contains everything. So DNA, the membrane, and everything that normal cells have, and plus the strange parts, which are the axon and dendrites. What are these? The axon is basically the output from this neuron, so it's how this neuron can talk to other neurons. And the dendrite is the input, so it's what other neurons use to talk to it. And generally, it's the case that neurons talk to other neurons, but this is not always the case. Because obviously, if neurons only talked to each other, we would not be able to interact with the outside world. And the reason why we can interact with the outside world, I remind you, is because there exist three types of neurons. So we have sensory neurons, which take the inputs from the real world. For example, we have some in our eyes that take light. We have some on our skin so that we can feel touch. We have some in our ears so that we can feel sounds, and so on for taste, smell, sense of balance, temperature, pain, and so on. So we have all these different kinds of senses, and the only thing that's different for those neurons is that the dendrites, which normally receive input from other neurons, for sensory neurons, they are made such that they receive input somehow from the physical world. So, for example, they might be excited by light or by specific chemicals that create taste and smell, or maybe they might be stimulated by movement, such as for touch feeling. And so basically there's three neurons. There's the sensory neurons that take input from the outside world and then use axons to talk to other neurons. There's motor neurons, which is what we use to move our hands, our legs, our bodies, and even our throat and tongue so that we can speak. So everything that we do in the outside world, including our all of our movement and all of our speech, is done by the motor neurons, which is basically the same structure, but the axon connects to a muscle, and we will discuss this in a moment. And then there is interneurons. Interneurons is just any neuron that receives input from neurons and sends output to neurons. So most of the neurons in our body are actually interneurons. And interneuron just means that here the dendrite receives data from other neurons and axon sends it out to other neurons. And by the way, if you're confused about these terms, I remind you, axon hillock just means the start of the axon, and axon terminal is just the end of the axon. So the cell body sends out an electrical signal from axon hillock. It travels all the way to the axon terminal, and there it triggers a release of neurotransmitters, which is how neurons mostly talk to each other. Now let's discuss what this is. This is called a spike. And a spike is a process through which this phenomenon, one second. Yeah, never mind. So spike is a phenomenon through which neurons talk to each other. And here's what happens. So we remember from our previous lectures that neurons all operate by electricity. And what we mean by that is that there are some charged particles inside the neuron, and there are some charged particles in our brain outside the neuron. So you can imagine that outside the neuron there is a water, but this water also contains some charged particles. And this difference of particles inside and outside neuron creates voltage. In this case, basically, that just means difference of charge. And neurons actually use that process. The way neurons use that process is that they receive the input by receiving charged particles, and this changes the charge inside the neuron. Here we can see on our y-axis we have neuron response, which is pretty much just the charge that builds up inside the neuron, and our x-axis we have time. So for example, in this case, the blue was some input that this neuron received. For example, let's say this neuron is the neuron on our skin, and it receives input every time we touch something. Like suppose we touch the cushion or we touch the spike or something. So first time we touched something, our signal started going up, but then we stopped touching, the signal went down. Then let's say next time we see another color, so it's another signal, like maybe this time we touch the pencil, then maybe we touch the pen, and then maybe finally we touch the blade. And you can see here slowly the voltage inside the neuron was building up from all the received inputs until finally it reached this threshold. This is called neuron threshold. And what happens is that when we reach this certain amount of charge, a lot of the gates will open inside the neuron and a lot of the changes will happen so that the charge will shoot up quickly. And this shooting up is called a spike. And this is why neurons can quickly, well, I mean, slowly build up charge, but then quickly send out a signal. And this spike is how all the neurons communicate, and this is the fundamental building block of communication in neurons. So again, the phenomenon is that it slowly builds up over time. If the input is not quick enough and not strong enough, then the charge dies out. But if a charge reaches this critical amount, it will shoot up in a spike and go to some maximum amount. After this maximum amount, it will cause a trigger of neurotransmitters in the next neuron, and then this one will calm down and die out all the way until it returns to rest. But actually, rest point is right here, but it goes a little bit below the resting point. What this causes is a refractory response, which basically just means that if this neuron fired right now, it will take a moment before it can fire again. And for that moment, it will have a decreased sensitivity. For example, you can think of this as if you just heard a loud noise, it will take a moment before you can figure out small noises after that. So you might not be able to notice small speech right after a loud noise. That is one way you could think of this phenomenon, roughly. So let's go into a little bit more detail here. And by the way, don't be intimidated by all the terms you see on the screen right now. We will break them down. So first, let's start right here at our time zero. This is our resting membrane potential. And by the way, membrane potential just means what is the difference of electrical charge from inside and outside the neuron. Because as you remember, the wall, which is the border of neurons inside and outside, is called membrane. So membrane is just border again. And membrane potential just means what's the difference of charge from inside and outside. So at rest, most neurons have a potential of around minus 70 millivolts. And this is where it's just resting, doing nothing. What happens after that is that voltage starts to build up as inputs arrive. And there is a critical point here. In this case, we can see it as a blue dotted line. And if it reaches there, a spike will be triggered. This spike is also known as hypopolarization because, yeah, basically it quickly shoots up and goes all the way into positive charge. So it started out as minus 70 millivolts, but it went up all the way into plus 40 volt, 40 millivolt around. The absolute peak at which it goes is called peak overshoot value, and it's called overshoot because it becomes positive charge, and that's, yeah, basically just a term called overshoot. And after a while, it calms down. This is a process of repolarization, and it comes all the way back down and enters this point. At this point, we can see the purple dotted line in this case is where the resting potential should be, which is from a point from which neurons are excited so that they can shoot up signals. By the way, if this is confusing, don't worry, we will see examples of this in a moment. But basically, once it comes back here, it actually goes even farther, and this is called after hyperpolarization. And this causes the refractory period we mentioned before, which is basically that for a moment, this neuron will not be able to be triggered again. And then it calms down again and goes all the way back to the normal state. So this actually happens very quickly. All this process that I described is just around 3 milliseconds, which means but this can happen more than 330 times per second. In theory, at least. But in reality, we will see that most of the brain waves are around 20 hertz, which means that most neurons don't fire more than 20 times per second, at least not in normal circumstances. Sometimes even less. The average is said to be around 1 times per second. But we will get back to that in our later lectures when we talk about brain waves. For now, all we need to understand is that this is a very quick process that happens in the brain, and this lets neurons communicate with each other. Now, how does this spike let neurons talk to each other? First of all, let's quickly describe the refractory period, and then we'll get to how the neurons actually talk. So, what we mean by refractory period is after the neuron has spiked and shot up and caused the release of neurotransmitters, and then it calms down and goes back down, it reaches down below the negative 70 millivolts, goes all the way down to about minus 85 millivolts. And this is a refractory period during which it's harder to trigger this neuron to spike again. And here's how this refractory period works. So we were at minus 70, and we spiked up when there was some input. Then we came down, and then we can see here the refractory period began. And so there is an absolute refractory period and then a relative refractory period. What that means is that when it's too low, the neuron is impossible to be retriggered, but after a while, strong enough input can retrigger it, and then eventually normal input is enough to retrigger it. Now, this might seem confusing right now, but after going through homeworks and some optional readings, you can build up more intuition about it. And actually, we will build up intuition about it in a moment. So far, are there some questions already in your minds? All right, we will see more clarifications of this because this is hard to grasp at first, but once you get the intuition behind it, it becomes clear just how the brain works. So now let's see an example of a synapse, which is, so basically here's the process. So neuron wants to send out a signal to other neurons. For that, electrical potential builds up inside the neuron, and when it reaches a critical level, it shoots up, causes a spike, which is just a lot of positive charge, and that sends the signal across the axon. The axon will eventually, through the axon terminal, arrive at a dendrite of another neuron. And where it arrives, that's where the synapse is. And let's see what happens inside that synapse now. So here's what will happen. We saw that some electrical signal arrived. This is the axon, the end of the axon, and this is the dendrite of some other neuron. So what was this process we're just looking at now? The process is, let's just restart it. Charge arrived. This caused some gates to open, calcium gates that we'll discuss later. Some ions, which means charged particles, flowed in, and it caused the release of neurotransmitters into the other neuron. This release of neurotransmitters is how neurons talk to each other, and this is where everything like our mood, our motivation, our emotions, and everything is regulated. So the neurotransmitters, which are these circles you can see that will be released right now from here, these red dots, will connect to the dendrite on the other neuron, and will open some gates and change the voltages such that those neurons can also spike. Then they will cause other neurons to also spike and so on, all the way until some neurons move our hands or move our mouth so that we can speak and move and everything. So this is a process that happens. And now to see more deeply what this process means, let's break that down. So first of all, what we're watching here is called a chemical synapse because the neurons talk to each other with chemicals, the chemicals that we call neurotransmitters. And here's the exact process. So imagine that this right here was the neuron that sent out the signal, right? So this is a cell body, these are the dendrites, and this one long tube is the axon. So in this axon, there was an electrical signal that was sent, and when it arrived here, it basically caused the release of neurotransmitters. And now what that means, all the bubbles you see here, these blue bubbles, are called synaptic vesicles. The synaptic vesicles are basically just bubbles that hold a lot of neurotransmitters. And when a signal arrives in the axon, these bubbles just dissolve through the exit and they shoot out the neurotransmitters in this opening. This space, the entire opening, is called the synaptic cleft, and it's where one axon shoots out neurotransmitters so that another neuron can take them in. And this process causes the communication between neurons, which is synaptic transmission. So you can see here, the axon terminals form the synapses with the dendrites or somata of other neurons. So let's break down that statement. This entire thing we see here is the axon terminal, so just where the axon ends, and it connects to a dendrite. In this case, this is the dendrite of another neuron. And this structure together is called the synapse. So when a nerve impulse arrives in the presynaptic axon terminal, and by the way, these are the terms. So presynaptic is the axon that is going to send the synapse, and postsynaptic is the dendrite that will receive this synapse. So basically, synapse has a sender and a receiver. The sender is called presynaptic, and the receiver is postsynaptic. So this whole structure is like this. And neurotransmitters are sent out from the axon and they bind. So they connect to some part of a dendrite and they use that to change the sensitivity so that signal and the potential that we see here can be more quickly or more slowly changed based on what neurotransmitters were connected. So we were talking about how the whole purpose of sending neurotransmitters is to make this process of spiking in other neurons faster or slower. So here's the process again. The charge arrives, some gates open so that charged particles can flow in. These particles will cause neurotransmitters to be released. They bind to the dendrite of another neuron, and then over there the same process repeats, which causes them to spike faster or slower. The neurotransmitters that cause the spike to speed up are called excitatory because they excite the neuron. And the neurotransmitters that cause them to slow down are called inhibitory because, well, they inhibit, so they slow down the process. And we need both kinds in our bodies because excitatory neurons basically make us, for example, more alert, more focused, more able to move and activate. And inhibition is needed when we need to, for example, relax, calm down, refocus our attention, or things like that. So they're both necessary processes, and our body makes sure to balance them out. Now, this was the structure of a chemical synapse. So basically, there's two kinds of synapses, and we'll get into that right now. The chemical synapse is when a charge arrives at the end of an axon, it causes the release of neurotransmitters, and then the other neuron gets these neurotransmitters and uses them to change how quickly its spikes work. And there is also another type of synapse called an electrical synapse. And this is a much simpler version of a synapse, actually. And here's what it is. So here in the middle, lower right corner, you can see, you can think of this as three neurons. So one neuron, another neuron, another neuron. And they are physically connected. So their membranes, meaning their walls, are connected. Like these two are connected, these two are connected, and these two are connected. And these connections are called connexon. And basically, they're just proteins that connect to neurons' walls. And because they are connected, when the connections are open, they build up charge at the same time. Because what does it mean to build up charge? So, in order for a neuron to spike, it has to collect a lot of charged particles inside it. But because these neurons are connected to each other with walls, they actually share these charged particles with each other freely because of diffusion. If you remember, diffusion is a process where particles freely flow between each other. Like if you pour water and juice together, they will quickly dissolve into each other, that kind of thing. And because of that, they will mostly have the same amount of charge. And so these three neurons will spike at the same time. This is called an electrical synapse because they are literally electrically connected in the sense that they share the charged particles directly. So the chemical synapse is when one neuron has to send out neurotransmitters to talk to another neuron. Electrical synapse is when they are directly connected, so the charged particle buildup happens in the three of them at the same time, and so they will spike at the same time. And the basic idea is that we need electrical signals, I mean, we need the chemical synapses that we see here with neurotransmitters when we need precision, like when we need to think specific thoughts or move a specific small part of a muscle or do some precise movements, or when we need to like form words. But if we need more general things, like if we just need to get excited or we just need to tense the entire hand, then we will use electrical synapses because they work faster and cheaper pretty much. So they have less detailed control, but they are more efficient and quick. So like if you have to harden all of your muscles in your biceps to lift a bag, for example, it will be done by electrical synapses. Now to quickly get an intuition of synapses, I want to show you this video from 2 Minute Neuroscience. And by the way, just a brief description. 2 Minute Neuroscience is a nice playlist where they explain some really good neuroscience concepts in 2 minutes, which is, I think, just crazy good. And I highly recommend this channel. I will link it in the description. Let's just watch these 2 minutes.
Where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss synaptic transmission. Most communication between neurons occurs at a specialized structure called a synapse. A synapse is an area where two neurons come close enough to one another that they are able to pass chemical signals from one cell to another. The neurons are not actually connected, but are separated by a microscopically small space called the synaptic cleft. The cleft is less than 40 nanometers wide. By comparison, a human hair is about 75,000 nanometers wide. The neuron where the signal is initiated is called the presynaptic neuron, while the neuron that receives the signal is called the postsynaptic neuron. In the presynaptic neuron, there are chemical signals called neurotransmitters that are packaged into small sacs called vesicles. Each vesicle can contain thousands of neurotransmitter molecules. When the presynaptic neuron is excited by an electrical signal called an action potential, this causes the vesicles to fuse with the presynaptic membrane and release their contents into the synaptic cleft. Once they are in the synaptic cleft, neurotransmitters interact with receptors on the postsynaptic membrane. They bind to these receptors and can cause an action to occur in the postsynaptic cell as a result. This action may involve increasing the likelihood that the postsynaptic cell will become activated and fire an action potential, or decreasing it. Eventually, the neurotransmitter molecules must be cleared from the synaptic cleft. Some of them will simply drift away in a process called diffusion. In some cases, the neurotransmitter is taken back up into the presynaptic neuron in a process called reuptake. Once back inside the presynaptic neuron, the neurotransmitter can be recycled and reused. In other cases, enzymes break down the neurotransmitter within the synaptic cleft. Then the component parts of the neurotransmitter can be sent back into the presynaptic neuron to make more neurotransmitter.
Right. So in this video, actually, he touched on another topic that we haven't discussed yet. And it's, yeah, right here. When he says that there are different things that could happen to neurotransmitters that build up between synapses. I mean, to be clear, most of the synapses that we will be discussing are chemical synapses, which means that axon sends out neurotransmitters and the dendrite of another neuron receives them. So most of the focus is on these kinds of synapses in all of the neuroscience study, and that's mostly what we will be focusing on as well. So here in this case, for example, what happens? So the axon receives some electrical charge. Because of that, the synaptic vesicles, these green bubbles, are forced to release the neurotransmitters that they have inside. And these released neurotransmitters now float here in this space called the synaptic cleft, and they sometimes randomly connect to the dendrites, which is this part of some other neuron. So after a while of floating, some of these neurotransmitters will be broken down by enzymes, and enzymes are just proteins that break stuff down or just generally speed up chemical reactions in our body. And some of them will return back to the axon so that they can be reused again and again and again. Now, generally, if neurotransmitters remain here for too long, that can cause a disease because that can mean that there was too much stimulation received by a neuron, and that might ruin the sensitivity of this receiving neuron. Similarly, if there's too little of neurotransmitters, then we might not be able to send signals, and this can cause, for example, loss of ability to move your hand if you can't send signals strongly enough. So actually, a lot of diseases that have, for example, paralysis are caused by weakening of this neurotransmitter release. So this is a very important process that we take for granted because generally it works just fine. And by the way, I will link this channel because I believe it's very useful. Their 2-minute neuroscience playlist is just wonderful. Now let's get back to our topic. Where were we? One moment. We were here. Let's just get back to where we were. Right, so we saw that there's a chemical synapse, which is when a charge causes the axon to release neurotransmitters, which the dendrite of another neuron will receive, and because of it, the charge will start to change on the other neuron and will cause the spike to happen faster or slower. And we also saw that there exists an electrical synapse, which is when neurons are directly physically connected to each other, I mean the cell bodies of neurons, and so the charge particle buildup happens in them together, and so they spike together. So these are the two kinds of synapses, but the chemical one is much more common and what we usually deal with. And generally, when you hear a synapse, it refers to a chemical synapse, not to electrical synapse. Before we go any further, do you have some specific questions? Let me see the chat as well. All right. We will repeat these concepts again, and as I will also send out a sort of brief reminder of everything we went through in this lecture so you can read through it in addition to rewatching the video as many times as needed. But yeah, of course. Do you mean this slide?
Yes, please.
Yeah. So this is the slide of electrical synapse. And here, let's break down everything that's going on here. So first, these blue things are just membranes of a neuron. So membrane is just a wall, border that every cell has. So here you can see, imagine these are 3 different neurons. And their membranes are connected, their membranes are connected, their membranes are connected. And these orange things are the connections, and they're called a connexon. They're made of 6 connexins, and basically because of the structure they have, they can be closed or open. So if they're closed, the neurons are not talking to each other, so nothing happens. But if they are open, there is an electrical signal between the neurons. And the electrical signal, I mean electrical synapse, which just means that charged particles can flow around here. And so if one neuron has a buildup of charge, that'll also transfer to this neuron and to this neuron. And so all 3 of them will build up charge roughly at the same time. That's what an electrical synapse is.
Thank you.
Okay. Yeah, and actually, everyone, I encourage you to ask questions. Partly also because if you're having a question, it's possible that someone else is having it and they're too shy to ask. So you could also be helping out someone else by asking questions. But I mean, I will also make sure to go through the topics again. All right. So we see that generally neurons send out their signals through an axon, and generally at the end, the axon sends out a neurotransmitter that another neuron receives. And that is the case for almost every neuron, but there is an exception. The exception is motor neurons. Motor neurons work slightly differently, and here's what they do differently. So motor neuron is just any neuron that ultimately moves a muscle, like neurons that let us move our fingers, our hands, our legs, our head, our neck, our, for example, throat and tongue, which lets us speak and also eat and swallow and everything. And what's important is that here's the difference here. So you remember that in a chemical synapse here, the axon of one neuron connects to the dendrite of another neuron. But in the case of a motor neuron, the axon of this neuron actually connects straight into the muscle. So the muscles have this motor end plate, which is a place where they can basically receive the signal from a neuron. And what happens here is that this kind of connection is also a chemical synapse, and the axon sends out a neurotransmitter called acetylcholine. Acetylcholine is a neurotransmitter that lets us move our muscles, basically, including the ones we use for speech. And so these axons release acetylcholine based on when we want to move our hands or fingers or legs and such. And this causes the motor end plate to get excited. And this excitement causes the muscles to contract. And the contraction of muscle is how we move. So we can see here, we will get more detail into this in Nini's lecture when she will explain movement. But general idea is that neurons send out a neurotransmitter called acetylcholine, which excites the muscles and causes them to contract, and that's how we move. So again, here you can see, for example, muscles are of course connected to bones. Each of the muscles receives input from some motor neuron, and here you can see these are the motor neurons. And so one motor neuron can give order to multiple muscles, but each specific muscle fiber only receives the input from one specific motor neuron. And again, we will also get into detail in this in our movement section. Now, one more time, let's go through what the spiking is. And here's what the spiking is. So we start, every neuron is by default at around minus 70 millivolts. That's called being at rest. Then as input arrives, we start to depolarize. And when we depolarize it around the level of minus 45 millivolts, certain gates open up, and this causes the depolarization to speed up. And it speeds up so much that it looks like a spike. Yes, Ekaterina? I'm so sorry, but I have different meetings somewhere else. Oh, the meeting will end in about 10 or 15 minutes, I think, Ekaterina. If you have to drop early, I guess it's fine as well, because you can watch the recording for the rest, so don't stress about it. But yeah, I think we'll end in about 10 or 15 minutes. Okay, let's see. So at rest, we are at minus 70 millivolts. Then as some input arrives, for example, because we see something or we are angered by something or we remember some memory at random, the potential starts to build up. And once it reaches the minus 45 millivolts, certain processes in the neuron start that speed up the increase in voltage until we get all the way up into the peak. At the peak, we enter repolarization. And in here, we have an absolute refractory period during which it's impossible to re-trigger this neuron because it first has to relax. It relaxes down all the way, and then it relaxes too much and undershoots, comes down to minus 75 or sometimes minus 85 instead of minus 70 millivolts. And then in here, it has relative refractory period, which means that you could excite the neuron, but it takes more effort. So, and then eventually the neuron will calm down and it will go back to rest. So this is the process that repeats in every part of our body trillions of times, literally each second, because we have about 100 billion neurons and each of them spike at least once a second on average. So it's like 100 billion processes each second. And yeah, this happens a lot. Now, let's discuss the specific types of neurotransmitters and what they do, because so far it's been an abstract idea, but we're actually very much familiar with these terms. For example, and by the way, neurotransmitters are also types of hormones. And hormones, by hormones, we just mean chemicals that can affect our body, our emotions, our balance, our behavior, and many things about us. And for example, let's talk about the four happiness hormones. So these are dopamine, oxytocin, serotonin, and endorphins. Roughly speaking, dopamine is for pleasure, but it's also for example, attention and focus. Oxytocin is sort of like warmth, affection kind of thing. It's secreted a lot, for example, during hugging. Serotonin is just happiness and self-satisfaction. And endorphin is excitement. So endorphin is actually the same as adrenaline. Adrenaline is basically a synonym of endorphin. So when you're excited about something, when you're hyped about something, when you're on a roller coaster, you have endorphins. So these are the different kinds of neurotransmitters, and they are all excitatory because they cause some actions or they build up energy in our body. For example, this is the structure of dopamine, and this is one of the most famous ones because, for example, overdose of dopamine causes addictions. Addictions like drug addictions, alcohol addictions, or even things like sugar addiction or maybe scrolling addiction, so that we are stuck to our phones, or maybe the addiction of watching too many movies or reels, or just anything that can feel pleasant and can give rewards instantly is abusing the dopamine pathway in our brain, and that's how addictions develop. So figuring out what to do with dopamine, one example being dopamine detox, when you intentionally deprive yourself of easy pleasures, is one way to fight addiction, for example. And there's some interesting research on this. For example, some people do dopamine detox such that they refuse to use their phone or to eat the delicious sauces and foods for a week. And this lets them reset their mind so that they can more easily gain pleasure and focus later. Because the problem with dopamine is that it also is needed for focus. So when you overdose on pleasure, you also ruin your ability to focus, which is very bad for work or study or just anything. These are the structures of different neurotransmitters. And I mean, I don't want you to actually memorize the chemical structure, but it is useful to understand what each of them does. So let's just go through them briefly. Adrenaline is for high levels of excitement, which is on roller coasters for hype, but in reality for like fight or flight situations. Like when you hear the sound of a lion or when you hear the explosion, adrenaline kicks in your body so that maybe you need to run away quickly and so blood needs to be pumped to your muscles, or maybe you need to prepare to fight, so you need to be sharp and alert, and that's adrenaline. There is also noradrenaline, which is for concentration, so when you need to focus on something. Dopamine is again for pleasure, but also focus and motivation. Serotonin is generally for happiness. GABA is a GABA is different from these ones because GABA is an inhibitor. What it means is that GABA is used to slow down or remove the spiking from the brain. So when we need to calm down, relax, and stop the brain activities, we use GABA. And by the way, if you think about it, some neurotransmitters have to be inhibitory because otherwise, what would happen when one neuron excites another, and this one excites another, and that one excites another, and then they keep cycling like that? If they never calm down, our brains would eventually fry because they would be always active. And so, to keep our brains balanced, there are some inhibitory neurotransmitters like GABA, which basically keep our brain down. They know how to calm us down, help us go to sleep, or just rest. Acetylcholine is for learning, memory, and also for movement. So every time we have to move our muscles, we use acetylcholine. And by the way, if you're confused how same neurotransmitter can be for learning, memory, and movement, this is because the same neurotransmitter will do a different job based on which part uses it. For example, in muscles, they are for movement, but in different parts of the brain, they can be doing different jobs. So yeah, same neurotransmitter will do a different job based on which part of the brain uses it. So you remember from previous lectures that we have, for example, a frontal lobe, a temporal lobe, parietal lobe, and an occipital lobe, and also some other brain areas. So each brain area uses the neurotransmitters slightly differently. Okay, let's continue. And by the way, I also write a bit about neuroscience topics, and you might find them useful for slightly deeper dives. And for example, here I have a small post about neurotransmitters. I'll link it. Basically, that they are in chemical signals. They can be amino acids, amines, or peptides. This is more chemical details that we may not need. We know that neurotransmitters work by opening ion channels on neurons. And let's actually discuss for a moment what ion channels are. So this is what an ion channel is. Here's what it looks like. This is an ion channel. So every time a neuron needs to receive an input from the outside world, the charged particles which cause the buildup of electric potential, it happens through ion channels. So ion channels are like these gates that let the outside world talk to the neuron. So in this case, this area is the cytosol and this area is the extracellular fluid, which means that so down here is the inside of the neuron and up here is the outside, just everything inside the brain. So these are like gates. So you can think of them like kind of like borders or airports between countries. So each ion channel will check, and for example, it will let calciums pass, but it will not let other materials pass. So the ion channels let specific chemicals pass, and this is how they regulate how much voltage will build up inside the neuron. And basically, when one neuron releases neurotransmitters into another, the neurotransmitters bind to the ion channels and they cause them to become more sensitive or less sensitive, or to open or to close. They have this somewhat complicated structure of how they work. You can read up more about it on this link if you want. I'll keep the link after the lecture. And the basic idea is that there are different kinds of neurotransmitters, so there's dopamine, serotonin, glutamate, oxytocin, melatonin, and so on. Many, many different kinds, but all of them work by changing the sensitivities on some ion channels on the dendrite of another neuron, which basically causes the spike to happen faster or slower. Now, how are neurotransmitters made? Neurotransmitters are actually made by the rough endoplasmic reticulum. This would go more into biology that we don't really need to cover right now. But basic idea is that you remember that neurons have a main cell body, and that's where neurotransmitters are created. And then they are carried to the ends of the axon terminal, and then they are released there. So there are different kinds of neurotransmitters, and we can talk about them. But the main part is that neurotransmitters then cause some processes on the other end of a neuron. Some processes that we don't need to get into right now. But the more important part is that this is how our brain talks to itself. And this is how everything happens. This is how we feel, how we think, how we move, how we can know that we exist, how we perceive things, and also, for example, how we love, how we hate, how we get angry or happy, and also how we get diseases and how we can cure diseases eventually. I will leave the links to all of this, but now let's return again to our slides. Let me just quickly find where we were. Oh, and yeah, that's pretty much it. And also, I recommend that you might like to take a look into this neuroscience readings that I have compiled here. For example, here we were mostly covering cellular neuroscience, like what is a neuron, what parts it's made of, and so on. And also, for example, how neurotransmitters work, how spikes work, and how synapses work. We saw that there's electrical synapses that literally directly connect the neurons, and chemical synapses that are axon releases neurotransmitters, dendrite of another neuron takes them, and because of it, it spikes faster or slower. We can read up more about them here. And also, for example, from previous lectures, we remember that brain has different parts, like frontal lobe, parietal lobe, temporal, occipital, cerebellum, and so on. So I think you might find these links useful, so I'll give them in the Discord group. And I guess that's pretty much it for today's lecture. This is a lot of material, so I'll try to make sure that the homework and also some additional materials reinforce it. I also highly recommend that you rewatch the 2-minute neurosciences video a couple times so that you can develop a deep understanding of it. And I also recommend that you try to sketch out how one neuron talks to another. So from the buildup of charge to spiking to sending the neurotransmitter to what that neurotransmitter actually does and how it works, and then how it gets cleaned up. So I highly recommend that you roughly sketch out each part, at least describe it in words and maybe even draw it if you like. I will include that in the homework. And yeah, that's pretty much it for this lesson. Now I want to hear the feedback or the questions from you. So for example, is there any slide you would like me to go back to so we can discuss it, or any specific questions or something? Okay, thanks for that comment, Ekaterina. everyone else. So here's what I'm thinking. I suppose we will clarify this more with the homeworks, but I'll just do a brief review of what we covered, and then we will end our lesson. Let me just briefly review the topics. So we remember that a neuron is made of a cell body, an axon, and dendrites. Axon sends out the signals, and dendrite receives them. And the cell body does DNA and everything to maintain this, basically. We saw that spikes happen when inputs arrive, it increases the voltage a bit, but then it decreases over time. Then a bit more increase, until we reach the threshold, after which we shoot up, and this is called the spike. And then we have to go back down to relax, and then eventually we can spike again. And this is the process that each neuron follows. We saw that this happens in around 3 milliseconds, and we saw that resting potential is at minus 70 millivolts. Shooting up happens at around minus 50 or minus 45, something like that. Then there's overshoot and repolarization, and sometimes we hyperpolarize afterwards, which means that we go even lower than minus 70 at like minus 85, which means that for a while it will take a stronger signal to cause this neuron to trigger again. We discussed refractory periods when we can't retrigger this signal, and then we can retrigger it, but after with some effort. We discussed the chemical synapses, which is that electricity arrives at the end of an axon, it causes a release of neurotransmitters as we see right now. They then bind to ion channels of a receiving dendrite, which then causes the spikes to happen faster or slower based on what's being received. And so that's the chemical synapse, like this. We also covered an electrical synapse, which is when neuron cell bodies are directly connected to each other. So whatever charged particles they receive, they share. And so they have the same charge at all times. And so they spike together. And we also covered motor neurons, which, unlike other neurons, their axon connects to a muscle. And this muscle gets excited, and because of it contracts, and that's how we move. And the motor neurons always use acetylcholine. So acetylcholine causes us to move, basically. And this is how muscles work. Each muscle is made of muscle fibers. Each muscle fiber receives the signal from one motor neuron, but one motor neuron can talk to multiple muscle fibers. So this was the spiking. So we are resting, then we are depolarizing, then we are repolarizing, and then we are going back to rest again. We also discussed some specific types of neurotransmitters. So dopamine, oxytocin, serotonin, endorphin for happiness, roughly. And that there are many different kinds of neurotransmitters for different jobs, like GABA for calming down, acetylcholine for movement and learning, adrenaline for fight or flight, dopamine for pleasure, and so on. And then we have more details available here. That's basically it for today's lecture. So if you get any questions, feel free to text later. That's not a problem either, or to ask right now. Either way is fine. And if that's all, I guess we can end our lecture now. Sorry, Salome, did you say something?
No.
Okay.
Sorry, I have one question for
Yes.
tomorrow's lesson. Sorry, we had a previous homework in Python, and we have homework for tomorrow. And you said that we would discuss this homeworks, and will we discuss the previous homework also?
Yeah, that's what I mean. So I gave the second homework, I think yesterday,
Yes.
but
Yes.
we will only discuss the first homework tomorrow, so that there's enough time to do the second one.
Oh, thank you very much. Thank you very much.
Yeah. Basically, I'm thinking that's the structure we will follow. So like next Sunday, we will discuss the second homework. The next Sunday, we'll discuss the third homework and so on, so that there's enough time for everyone to do the homework. Because I don't want the deadline to be strictly tomorrow. If you don't have it done by tomorrow, I want you to still be able to keep working on it, basically. Yeah.
Okay, thank you very much.
Yeah. Okay, any other questions? All right, then. Thanks everyone for attending, and I'll see you tomorrow.
Thank you and goodbye.
Thank you.
Bye.
Goodbye.
Bye.